Okay, lecture 3, we'll talk about the characteristics and types of parenteral injectable drug dosage forms. Okay, so there are a lot of characteristics or requirements of parenteral drug forms. Okay, the injectable drug forms. Of course, the most important requirement or the characteristics of the parenteral drug should be the safety of the drug. Okay, so safety can be defined as its freedom from adverse toxicological concern. What it means is it should be safe when it's injected to the body of patient. Okay, there's no toxic substance, there's no severe side effect or adversary effect, so there's no harm done on the patient when the drug was given as injection. That's what is most important, right? So since it's directly injected to the body of the patient, so it should be safe. Makes sense. So when selecting the excipients, so active ingredient is mixed up with the excipients, right? That's a compounding. Okay, so the API should overcome drugs' solubility, stability, toxicity, and delivery. The formulation scientists should carefully follow the safety requirements. So when you consider the best formulation for your drug, always safety concern is number one. That's what it means. So remember the Kiefer-Harris amendments, okay? The FDA, the law, the Cosmetic Act, and the pharmaceutical preparation should be tested for safety in animals, okay, animal study, as well as the clinical trials. So safety is very important. Number two requirements, number two characteristics would be sterility. Since it's injected into the human body again, your drug should be free from any microbial contaminations or contaminants. Okay, so it should be sterile. That's what it meant. So your manufacturing process should be a very clean and sterile procedure. Okay, so you should provide a sterile process, maybe aseptic filtration, if that is what you need to do. Since you're dealing with the biological molecule, you have to make sure that that's a validated process. And you have to do all the manufacturing under the clean room environment, of course. And the aseptic processing, all the steps should be validated. Okay, training, application of good aseptic practice of individuals should be done, of course. And sometimes you need to use antibiotic preservatives in your drug. So we'll talk about formulation, what things will go in there. One of the excipients that are needed may be added sometimes if especially your drug is a multi-dosage form, and you have to add antiviral, antimicrobial preservative chemicals that can inhibit the growth of bacterial cells. So that is very important. Okay. Number three requirements, non-pyrogenic. So we talked about endotoxin. We will even do the lab on LL assay. So pyrogenic is a very toxic substance that can cause the death of your patient if it's mixed up with your API in your drug, injectable drug, right? So your drug should be free of endotoxin, pyrogens. Okay, maybe it can have some, very little bit, but not a lot for sure. Okay, so again, pyrogen endotoxins are fever-inducing entities. Okay, that should not be there in your injectable drug. So how do you measure the presence or absence of endotoxin? LIL assay is the test method. Okay, we talked about it. So in your manufacturing process, there should be various subtypes of depyrogenation, meaning the ways to remove pyrogens. Okay, depyrogenation method should be included in your maybe cleaning or manufacturing processes. So cleaning is a big part of depyrogenation, of course. Okay, and then time limitation of any open container, and validated depyrogenation cycle for glassware. So normally it requires a high heat treatment. That is what it is. And validation of pyrogen endotoxin removal from rubber closure system like stoppers and things. And how many times you need to wash and rinse. And water system. What water do you use? The required water is called UFI, water for injection. Okay, you have to use endotoxin-free raw material, so endotoxin should not be there. So non-pyrogenic is another requirement that's important. So you've seen this, right? So LAL, how it's collected from horseshoe crab, and then this is the LAL assay, the color-based assay, quantification assay. And then what is this? It's an oven that can heat the pyrogenate the toxin 6, like very high temperature, like 650 degrees, in just a few minutes, 1 or 2 minutes will be good. So we'll talk about depyrogenation methods, different kinds later on. So understand, baking is one method of depyrogenation. Okay. And then another requirement or characteristic is particle free. Okay. So since your drug is injected into a lot of cases, maybe through the blood vessels intravenously, the particles should not be there. If you have particles, what's going to happen? It can clog up your blood vessel, which is not good, right? So freedom from visible particles is a requirement. So what do you see from this picture? This is the vial inspection, the visual inspections. This is an inspector. He's going to hold a couple of drug vials with the liquid drug in it. He's going to maybe swirl the vials and look at the vial under the black background, white background, and try to see if there are visible particles in there or not. So the particles that can be detected through naked human eye are about 10 or 25 micrometer or larger. Okay, if particles are smaller, then they are not visible. They are considered subvisible. Okay, so there are different ways of detecting particles, but your drug should not have a lot of particles. That's for sure. Okay. So how do you remove, or how do you make sure that there's no particle going into the drug? So of course, valid cleaning method is important. Your vial should be all cleaned up, and all the equipment, packaging materials, valid solution filtration. So filtration is a good way of removing any particles. And then adequate control of production. Of course, the particles should not be in your air, clean room air either. And testing environment, so that's why you're measuring the particles with the particle count all the time. And then adequate training of personnel. A lot of particles can are generated from individuals, people actually. People are terrific shedders. They drop, shed a lot of skin flakes and all those things, hair particles and all those. Okay, and the testing and using sterile product solutions and employment of required compendial testing procedure for detection of both visual and subvisual particles. So make sure your drugs should not have particles. Number 5 characteristic or requirement is the stability of your drug. So a lot of cases your drug is made out of biological molecules. Biological molecules are very sensitive to heat, oxidation, the fluctuation of heat, and sometimes air as well. And so pH, okay, or a lot of salt, too little salt. So many physical conditions affect the stability of your drug. Okay. So your drug should be kept stable. That's why you're compounding your drug with the excipients, and that's why you have to keep your drug maybe cold, refrigerated condition, or sometimes freezing condition. Okay. So parenteral drug should be stable under predetermined manufacturing, packaging, storage, and usage conditions. So your drug should be tested to make sure that it should be stable under manufacturing condition as well as the shelf storage conditions. Chemical and physical stability should be maintained throughout the shelf life of the product. Shelf life, what is that? How long you can store your drug after your manufacturing before it's used in patient care. Okay, how do you determine the shelf life? Normally it's tested by your formulation scientist or PD scientist, process development scientist, and they see and how long of the time is required for your drug to lose its probably structure and stability and safety, about 90%. That determines the shelf life actually. Okay. So complicated chemical structures and vulnerability to environmental conditions such as temperature, light, even light. Some biological molecules are susceptible to a strong light. They lose their structure and efficacy. pH, shear, physical force, metal impurities. Some of your drug may interact with metal, okay, which is not good. Oxygen, of course, causes oxidation, okay. And the therapeutic peptides and proteins offer enormous challenges because they are very prone to damage by these physical factors. So that's why your drug is compounded with excipients, mixing. But compounding process could be a stressful process to your drug. Mixing, filtering, filling, stoppering, sealing, storing, shipping, and handling, they all affect the stability of your drug. Okay. Many parenteral drugs are unstable in solution, so that's a challenging process. That's why sometimes you have to freeze dry your drug, lyophilize your drug. If you remove water molecules from your drug, it may be stable much longer period of time. And the maintenance of sterility throughout the shelf life and usage also affects the stability of the drug. Okay, you have to keep them sterile for a long time. And then comparability of your drug. So when you formulate your drug, when your drug is mixed with excipients, okay, those added substances cause any instability. So that's the stability we're talking about. They should not. Packaging materials should not interact with drug. They should be inert. That's part of the requirements, like vials and whatever. And other diluents. Okay, if you're adding maybe solvent before your drug is used. For example, if you have a lyophilized drug, you need to add sterile water, UFI water, before it is used. Okay, and then is your drug comparable for that process? Okay. So, in many cases, your parenteral drugs are manipulated before injection, meaning they are treated with some process. For example, lyophilized drug is reconstituted by the sterile water, UFI water. Okay, is that process comparable? Does it, may cause any harm to your drug structure? We're talking about this. Okay. And especially large volume drugs, infusion fluid may contain more than one drug already. Okay, so are those drugs comparable to each other? Okay, so something you need to test in terms of the safety of your drug. Another requirement is the isotonicity. Isotonicity, what does that mean? So your drug is a biological molecule normally made by a biological system, and biological systems have certain constant condition. Okay, of course, number one is what? There's plenty of water in the biological condition, like cells have 70% mass made up with water. Okay, and then besides water, it has a lot of salt and some other physical conditions. Okay, so you need to provide a very similar condition as a living cell. So your drug molecule should be stable. So that's isotonicity. Among the requirement is a salt composition of the solution should be a certain degree. Okay, so if the salt concentration is changed, okay, your drug molecule will be damaged. Okay, or if your drug is too salty, meaning it has too much of salt for some reason, so if your drug is injected to the human body, so a lot of extra salt coming in with your drug may cause some harm to the human body. Okay, so that's what you're talking about. Isotonic, meaning hopefully your drug should have a similar salt concentration as the human body. So when it's injected, it's not cause any of these effects. So hypertonic drug, meaning what? If your drug has a lot of salt with it, so if it's injected, increased amount of salt going into human body will cause this, right? Because your cells will be losing water, because water will migrate from the cell to outside. So they will die through shrinkage. Or if your drug doesn't have enough salt, and your drug, when it's injected, is sucking up the salt, okay, then what? Since the water molecules will move from your drug side to the cells, your maybe cells will be expanding and they may rupture. So that's a hypotonic condition. So ideal condition is this: your drug should be isotonic. Okay,